Hi all, welcome back. Okay, last time out we talked about red blood cells or erythrocytes and blood in general, like the makeup of blood, plasma, white blood cells, red blood cells, platelets, that kind of stuff. This time out we're going to talk about white blood cells, which is a major portion of the immune system. Right? And so white blood cells, which sometimes are just called leukocytes or leukocytes, right? Often abbreviated with WBCs, like red blood cells, RBCs. And so often you'll see that on forms and things like that that you might need to fill out is what's the RBC count, what's the WBC count, um, instead of writing it out. So their main purpose is to protect against foreign invaders, diseases in general. Okay, and so when we talked about red blood cells, we said that the average red blood cell lasts about 120 days. I'm not sure if I talked about this, but that's an average adult red blood cell. Juvenile or young individuals, their red blood cells are not in circulation that long. Normally, maybe 60, it depends on the age of the individual. 60 days to kind of 90 days, maybe for young individuals. If you're an infant or newborn, it's less than that, maybe. And that has to do with just the body's moving a lot of blood, growing, things like that. So the blood is being circulated a lot more often and into spaces, and so it gets damaged, used up. A little bit different size also. Red blood cells in individuals that are younger are bigger than red blood cells that are in adults, but nonetheless, about 120 days when we talk about being an adult. Leukocytes, on the other hand, pardon me, have a very different kind of lifespan, and it really determines what type of white blood cell are we talking about. And so we'll come back and talk about kind of the lifespans in general between these in a second here. Again, like the red blood cells, they're produced in the bone marrow. Hormones are going to trigger this, okay? And then after that, they can be differentiated in different regions of the body, okay? And so sometimes they're mature coming out of the bone marrow, sometimes they're immature and they need to go to a different region to be differentiated or mature in a different region of the body. Okay, so there are 5 types of white blood cells, 2 main categories. The first is the granulocytes. Okay, the granulocytes will have granules, enzymatic granules within their cytoplasm. These granules are used to destroy proteins, typically. Can destroy lipids and other things, but these are enzymes, protein enzymes that are released to destroy foreign bodies. Okay? And so when we talk about neutrophils, eosinophils, and basophils, for the most part, all of them are going to last anywhere from a couple of days. Neutrophils have the least lifespan of the granulocytes, but in all cases, we're talking a couple days to maybe a week. So their lifespan's really short. In some cases, if you have a bacterial infection and you have a lot of neutrophils, their turnover might be not days but hours, and so a lot less circulation time than red blood cells. And then you have the agranulocytes, or the non-granular white blood cells, which means that they may have enzymes, granules inside their cytoplasm, but they're not noticeable. They're smaller, much smaller, and so they have, from a visual perspective, they look like they don't have any granules, and these guys look like they do have granules, and that's where the separation between the two comes in. And you have lymphocytes, which we'll dive into a lot more. And you have two types of lymphocytes, T cells or T lymphocytes and B lymphocytes. And then you have monocytes. Again, lifespan here, a little bit longer than the granulocytes, but it depends on what white blood cell we're talking about. Some lymphocytes, like memory B cells, they can last years, 10s, 20s years. So you can be, subjected to chicken pox when you were very young, like 5 years old, and still have memory cells to that virus when you die. So you can have them for a very, very long time. Same thing with T cells, they can last a long time, and depending on the T cell. Monocytes typically they last a little bit less, maybe up to a month in the system. Okay, we'll cover what each one of these, the purpose of each one of these as we progress. So what's the function of the white blood cell? Okay. Diabetes, the white blood cells tend to have the capability to squeeze between cells in the capillary wall so they can leak out of blood vessels. This is really important from the perspective of if you, in your connective tissue where you have blood vessels running through your connective tissue, if you have a an infection or a site where foreign bodies have made it in, the diabetes or the white blood cells will squeeze through the capillaries, push their way through kind of the connective tissue there and into the region where the infection might be. So it's really like they leak through your capillary walls. Okay, in general, they have proteins that can adhere to sites of injury, and so not only do they have the capability to destroy foreign bodies, but they can also help with the clotting process. And we'll go more into this when we talk about how clotting works. Most of the time, when we're talking about white blood cells, we're talking about their ability to remove things that are foreign or damaged. Okay, it doesn't have to be foreign. It can be damaged cells within inside your own body that are self-made. They often get what we call phagocytized, so they're engulfed. So if you remember back from like previous courses and previous lectures, okay, they wrap their membrane around the foreign body and bring it in as a vesicle, and then they can dump digestive enzymes into that vesicle and it destroys the material. Normally we're talking neutrophils and monocytes that are the active phagocytes that occur in the white blood cell or in your blood. Excuse me. Other ways at which they can react, they can reduce the spread of infection by closing off regions and in enhancing or tricking, basically enticing your body to push fluid to that region. So if you get like a splinter in your finger or a cut or something like that, you'll get inflammation in that region, swelling, because fluid will be pushed to that region. Okay, and it's really increasing the permeability of the capillary beds. So it's pushing a lot of fluid there. A lot of this will be white blood cells that are associated with it, but they can also secrete different proteins and have different reactions like histamines and hairpin and things like that, which can cause swelling and retaining of water and whatnot. Okay, again, they have this ability to attach to infection sites, attached to foreign bodies, attached to damaged cells, and we call that positive chemotaxis. Okay, so they're attaching to the chemicals that are being released by damaged cells or inflammation that's occurring in that region of the body. Okay, and we'll look at this in more detail in a little bit. Okay, so this is the diabetes. Okay, this is the ability. So remember, we're talking about blood capillaries. We're talking about these hollow tubes that are carrying blood. That blood, I mean, here it's just showing you white blood, but obviously there's red blood cells and plasma within that capillary. But that diabetes is really the leakage of white blood cells into the connective tissue. So you might have a splinter that came in and it's stuck in the connective tissue, and maybe on that splinter was some bacteria, and so you get these leakage or white blood cells that are escaping the capillary beds and into the connective tissue to fight those foreign bodies. Okay, here's another example of kind of what we're talking about. Again, a splinter is it's a good example. This could be anything. It could be a cut. It can be, a scrape. It could be anything that's going on. It could just be like you inhale a bunch of viruses, you get bacteria in your mouth, whatever it might be. Okay. That bacteria is going to make it past the first layer of defense. The first layer of defense, we talked about this a while ago, is the epidermis. That epidermis, especially the corneum layer of dead cells, really provides that first layer of defense. If that's penetrated, and now, so this splinter is going down into the dermis, right, where the capillary beds and the blood vessels are, now you can have the reaction to the foreign bodies that might be on that splinter, in this case, maybe bacteria, right, is now in a region where that bacteria can start consuming material. It can start irritating, consuming cells, eating away at nutrients and things like that that might be in this region. So then you can have leaky blood vessels, which are releasing, in this case, neutrophils. Neutrophils are the white blood cell that destroys bacteria. So it's going to leak out neutrophils into the system to destroy, phagocytize this bacteria. And so it'll engulf the bacteria, break it up, and often it will present it as pieces of protein and lipids and things like that to the learned immune system, T cells, B cells. We'll come back and talk about how that works. Okay. All right. Talk a little bit more about the functions and kind of what they look like. So neutrophils, again, they are the most abundant white blood cell, and normally they're reacting to prokaryotic invasion. So if you have a bacterial infection, an archaea infection, if it's prokaryotic related, neutrophil numbers will increase, and they're what's typically phagocytizing bacteria and archaea. Okay, they might also increase in numbers when you have a viral infection, because viruses are often using bacteria to replicate. So when viruses enter the body, they're using native bacteria that are also in the body to replicate the virus. And so you'll often see neutrophil numbers increase with viral infections also. Okay. And so normally when we're looking at them, we're looking at the lobed nuclei. Okay, so sometimes the nucleus can be in two separate sections. Sometimes some of these sections are connected. I mean, sometimes it can be five sections, what looks to be five separate nuclei. But you're going to have small granules, and these small granules will typically, under most stains like a right stain or something like that, they'll dye kind of purple, but they're smaller granules than you expect to see in uh, some of the other white blood cells, the granulocytes. Okay. The eosinophils, right, they are more related to and going to react to eukaryotic uh, foreign invaders. So if you have parasitic worms, if you have malaria, trypanosoma, um, if you have giardia, a lot of these are protozoans or even animal-like parasites. Often you'll get a reaction to those from the eosinophils. Okay? Their granulocytes are much larger than the granulocytes and neutrophils, but again, they can have that bilobed nucleus. So these two, their nuclei can look very similar to each other, okay, but the granules are very different. So the granules are much more coarse, okay, and often will kind of dye either a pinkish reddish color or even white. It depends on the stain that's used, but again, the granules are much larger. Okay, back to neutrophils real quick. So normally we when we're looking at neutrophils and we're just looking at white blood cell counts, somewhere around 60% of the white blood cells are neutrophils. So they're the most abundant. And then eosinophils probably somewhere around 1% is very typical, though I mean obviously if you have a infection from a eukaryote, so if you have giardia or malaria or something like that, you might expect to see these numbers much higher. And so also allergic reactions to things, especially if they're fungal based or if they're based in like pollen or something like that, where it's a eukaryotic cell that your body's reacting to, again, you'll see higher levels of eosinophil. So normally, when we're talking about neutrophils, we're talking about prokaryotes. We're talking about eosinophils. We're talking about eukaryotes. It's not always the case, but it's fairly typical. Excuse me. All right, the basophils. Okay, so basophils, similar to eosinophils with large granules, but these granules under a normal acid stain will typically stain dark purple or darker than the nucleus. Also, another thing is often the granules will be covering the nuclei. So sometimes it's hard to see if the nucleus is bilobed or what's going on. But big granules cover it, can get confused with the eosinophils because they're similar in size, similar in shape. Again, it will depend on the stain that's used to make sure that you can differentiate between eosinophils and basophils. Okay, most of the time these are going to react to damaged cells. So when you have something like a cut or a puncture wound or something like that, it'll often stimulate inflammation in that region. They can do so by releasing heparin, and they are involved with clotting along with the thrombocytes or the platelets. And so not so much reacting to eukaryotic or prokaryotic foreign bodies, but more reacting to damage to cells. So if you have a lot of damage that's going on in the cells, the basophils will migrate to that region and try to mitigate that damage by clotting off um, blood vessels, capillaries, things like that, and then also increasing inflammation and just uh, making other cells come to the region. If it's a bacterial infection due to the splinter, these guys might be, making sure that neutrophils are coming to the region. So they're stimulating the production of neutrophils and, getting neutrophils into the region to take care of those foreign bodies. Monocytes, these are the largest. They are non-granular, or yeah, I mean, in general, under a scope, the granules are very hard to see. Um, Back to the basophils real quick. So maybe 1%, somewhere in there. So eosinophils and basophils will often have similar numbers, around 1% of your white blood cells. Monocytes maybe a little bit more, maybe around 3%. But again, it depends on the infection. It depends on the individual uh, and whatnot. But in all cases, neutrophils are by far the most abundant. And then lymphocytes, which we'll come to here in a second. The monocytes are the largest white blood cells. Okay, four to five kind of red blood cells big. So you could stick, 4 red blood cells inside the cell. So it's about double the size of a basophil. No real granules. The nucleus can look bilobed, can be disjuncted, but it's often not separated. And their job is really phagocytize things. They phagocytize your self-made cells. If your red blood cells are damaged, they eat them. If there's bacteria in the system that's not taken up by neutrophils, they eat it. If there's a parasitic worm, if there's trypanosoma, if there's malaria inside a red blood cell, it'll eat it. These guys are the generalists of the cell. Their job is to just go around and eat things. And then they present that material to the learned immune system, the lymphocytes. Okay. All right, so the lymphocytes, okay, smallest white blood cells. They're agranulocytes, so they don't have granules. And then I kind of already touched on this. There are 2 types. You have T cells or T lymphocytes, and you have B cells or B lymphocytes. Okay, and these are both based on learn immunity. So the other cells, whether it's a neutrophil, basophil, eosinophil, um, they're all what we call innate immune system. So they belong to your genetically controlled immune system. So they're going to react to things that enter the body, things that foreign bodies, even self-made things that are malfunctioning. They're going to react to them basically the same way every time. T cells and B cells, on the other hand, they're going to learn from previous infections. So this is how immunity works when you get things like an immunization shot. Okay? The idea is to trigger T cell, B cells, so that if you are subjected to that material at a later date, you already have memory of it and you can build up a quicker response. And we'll talk about how this works here in a little bit. So again, excuse me, about 25 to 30% of the white blood cells. So neutrophils are the most abundant, and then probably lymphocytes in most cases, and then followed by your granulocytes and whatnot. These guys are the longest lived, potentially many, many years, depending on what white blood or what lymphocyte we're talking about. Okay. So normally one of the key components of lymphocytes is that they're just a little bit bigger than a red blood cell. And the nucleus takes up the majority of the cytoplasm. So here you can see the entire cell, and that nucleus is really large, and there's not much cytoplasm. That's normally a real key feature to distinguishing lymphocytes from other white blood cells. All right, when we do white blood cell counts, okay, most of the time anymore, if you work in a facility that has any amount of money, these are automated. Just a drop of blood's placed in, and it's just based on the reflection off of the white blood cells, and so it'll do an automatic count for you. But if you're working in a clinic that may not be using automated counts, you'd use a hemocyameter, okay? And this is just going to be basically a slide that you put blood on, a known amount of blood on, and you're gonna have some grids there, and then you're gonna count white blood cells in those grids. Most of you have counted white blood cells before. It's just like that. You just count them, differentiate what the different white blood cells are, and you can get total numbers of white blood cells per microliter of blood. You can get the number of lymphocytes, the ratio of lymphocytes to granulocytes. You can get all kinds of things that might tell you if the individual is sick or what might be going on with their white blood cell count. So a couple things. If these blood counts are greater than about 10,000 or 10,500 microliters per blood, it would indicate that there's an infection. And so normally, unless this is a super athlete, unless this is like an individual that is training for marathons or training for the Olympics or is a professional athlete, that their numbers might be over that just because a lot of white blood cells are removing foreign bodies. And so if you're constantly burning through heavy amounts of food and glucose, you'll produce a lot of radicals. So if you're Michael Phelps and you're eating 12,000 calories a day and burning through every bit of it, then you might see a leukocytosis or high white blood count because it's removing a lot of those foreign bodies and damaged red blood cells, etc. But it also, in most people, it would indicate that you have an infection, okay, a bacterial viral infection, or potentially a eukaryotic parasite, a fungal infection, or parasitic worms, whatever. Leukopenia is the opposite, low white blood cell count, less than 3,500. Okay, this might be associated with things that may affect the white blood cells, or the white blood cells are being used up and not replaced. So anemia, maybe you don't have enough iron or blood cells in the system. Maybe you have viral infections that might be deteriorating or changing often enough that your white blood cells can't keep up. Okay, the flu, other things that basically your white blood cells can't be made as fast as they're being used, and so you'll have white blood cell counts go way down, and with certain types of viral infections especially. And then, of course, the knowledge that we can get from a differential white blood cell count would tell us a little bit about what's the potential infection. Okay, so if you see high levels of neutrophils, you might know, okay, we have a bacterial infection. If you have high levels of eosinophils, you might say, oh, this is a eukaryotic infection. So this individual's got maybe a parasite. Could be a fungal infection because those are eukaryotes. Could be related to, just in general, like hay fever or something like that, where they're reacting to pollen from a eukaryote. But often when we see things like people who consume water that's not filtered, and maybe they pick up Giardia, which is a protozoan, and they get really sick, they'll have really high levels of eosinophils to try to deal with that. Okay. Other things, like if there's a lot of white blood cells in ratio, so maybe you have lots of neutrophils and very, very few lymphocytes, okay, it could be related to something, maybe in some cases like an HIV infection, or opposite, maybe you have huge numbers of lymphocytes and very few other white blood cells, it could indicate that you have cancer, like leukemia, where you're producing massive numbers of lymphocytes. Okay, so let's look at some clinical applications that are related to that. Like I just mentioned, leukemia or cancer of the white blood cells. Um, so normally this is classified by basically the speed at which the onset occurs. Okay, so if you have acute, they appear suddenly, progress rapidly, or you have chronic, this is slow, and could be undetected for many years even. It also determines where is the cancer, like what type of cancer it is. So remember, you have two types of white blood cells. You have granulocytes or you have lymphocytes. Okay, if it's based on the lymphoid stem cell, which gives rise to B cells and T cells, then it's probably cancer that's occurring in the lymph nodes. Okay, and that's a lymphocyte problem. Okay, if it's a myeloid cancer, then it's probably a cancer that's occurring in the bone marrow. Okay, and that's a granulocyte problem. So the way this would look is if you have huge numbers of things like lymphocytes, you might have a lymph node cancer. If you have a large number of, say, neutrophils or basophils or eosinophils that you wouldn't expect, you might have a bone marrow cancer. Okay. So excess number of white blood cells can do lots of things. Remember, some of these white blood cells, their job is to create inflammation. So the basophils, they're creating more fluid to flow into regions. And if it's flowing into regions and you're building up lots of fluid, you can get headaches, you could get nosebleeds, you could produce fevers because it's attracting material to that region. All these things can occur when you have high numbers of white blood cells, typically myeloid cells. Okay? Again, how we treat this, it depends on the country. It depends on a lot of things. It depends on which type of cancer you have. If it's related to the bone marrow, you might have a bone marrow transplant. Okay, the bone marrow is removed, stem cells transplanted back in. If it's related to the lymph nodes might be removed, especially if there's tumors that are present in the lymph nodes. Lymph nodes might be removed, and then you try to replace lymphocytes, and so the individual might need blood transplants more often. Okay. Of course, chemotherapy is going to slow down rapid cell growth, things like that. There's lots of ways at which we try to treat these, but leukemia in general it's a cancer that has a high death rate because these cells are related to your immune system, and if your immune system's broken, then maybe the leukemia is not going to kill you, but an infection from bacteria or a virus might. And so that's where that really comes into play. Okay, so looking at something like someone with leukemia, myeloid leukemia. So here you can see normal counts. You have, some eosinophils. You have a lymphocyte. Okay, but just normal numbers. Everything looks pretty normal in this cell. And then you jump over to an individual with myeloid leukemia and look at the number of neutrophils that they have in this system. Okay, they got quite a few lymphocytes also, big basophil, but lots of neutrophils, far more than you would expect. It's almost the same number of white blood cells as you do red blood cells. Remember, red blood cells are about 95% of the blood should be red blood cells, with far fewer white blood cells. So this is one way that you can tell. Another way you could tell is, remember when we looked at those hematocrit levels, and you take and you spin someone's blood down, and the plasma is about 55% and the hematocrit's about 45%, with you know, the gray layer or the white blood cell layer is like less than 1%. If that's more like 4 or 5% or 6%, okay, then something might be going on. This individual's producing way too many white blood cells than you would expect by chance.